that point in the year. Now, you guys don't have any questions, you know what you're doing. All good. Sunny, or you haven't even studied yet. I don't know. I don't wanna know. Getting there. Urinary's a tough one. Remember, just took it. It's fair. It's fair. Let's just go over it. See if you guys can put it together. As I was taking it, it didn't take me very long to take. I was just kidding. But, it didn't take me very long, but I've got a pretty good chemistry background. So for me, it wasn't tough, but there'll be some parts you're gonna have to know. I'm gonna go through it today, but you'll do fine on most of it. It's about 40 multiple choice, 12 matching-ish, and then there's your analysis. So I've got a patient, and I listed out just like we did in here on Monday. Gave you the results of the urinalysis, and you have to give me names of the disorder, like albuminuria, pyuria, hematuria, whatever the problems are, and then give me a possible diagnosis. I certainly know what specific gravity is and how it's relevant to osmolarity. Those are all important topics that are on the exam that you'll be assessed on Monday. So it'll be at 8, then we'll have slides. We'll have 2 models in back, identification on the model, and you'll be out of here in less than an hour, I'm guessing, on Monday for our 3rd exam. So let's start up here with the review slides. As we start, 3 slides that you need to be responsible for. Beautiful picture posted on Moodle. We're going to go through that as well today. But out of the 3 organs that we are assigned, which one's this one? Ureter. Good. Okay, ureter. Lined with? Transitional. Good. Middle layer? Smooth muscle. And the outside? Serosa or adventitious shell, except either one for that. Good. Something relevant on the multiple choice came out that how does urine actually move through the urethra? I'm sorry, ureter. Peristalsis. So that's how it actually moves. So remember that as a possible answer for a question is peristaltic movement in the ureter actually moves the urine through. And so that answers the next question is fluid. There is a matching section on this exam that lists out 5 different structures related to the kidney and kidney system, and you'll have to identify whether it's plasma, urine, or filtrate. And this would be blank. Urine. That's urine. Good. Normal pH for urine? 5 to 8. Specific gravity for this exam, I accepted I put in a big range. Taking everybody in the world, normal specific gravity. (1.001 to 1.035) is a normal range for specific gravity. When you talk about osmolarity, is this a high or low osmolarity? Good.
Low.
There you go. Make sure you figure that out. There's a couple questions on osmolarity. Low osmolarity means it has a low number of solutes in a given sample of solution. High osmolarity means that it has more solutes in a given sample of solution. Which one? What's this color of this? Pale.
Lighter yellow.
Pale or clear? And this would be a darker yellow. It all came out of the lab and it's on the exam in a couple places, so make sure that you understand specific gravity, osmolarity. Yes?
So with the ureters, there was transitional lining, smooth muscle, and what?
Serosa. S-E-R-O-S-A. Outer, this is the outer layer. And then I'd like you to know that it has urine, and we move urine by peristalsis. That all comes off that slide. Organ. Kidney. This one's complicated. All right. Structure. Glomerulus. Structure.
Capsules.
Capsule. Cell type. Macula densa cells. Type of receptor. Chemo or osmo receptors. I accept either one. They both mean the same thing. Osmo or chemo receptors. Cell type. JG cells. Type of receptor. Mechano or presso. Very good. Mechano or presso receptor. They measure the pressure. JG cells release blank. Renin. Yeah, we posted on the forum last night, yesterday, or Monday, when was it, Monday? Yesterday. I listed the chemicals that are on this test with release point, target tissue, and function at the target tissue. It's pretty well filled out. There's a couple corrections, small, and some things that need to be filled out, and I ask you guys to do that as a group. I would expect by sometime this weekend that that's all filled out and completed by you guys. I'll check it Sunday, get it back, and make sure that you guys are on the right track, but I'm not gonna do it for you. We're in AP3 now, and you guys know how to study for this class. I'm trying to teach you how to study for the rest of your life, is that this is how you do it, and it takes a little bit of work, and I don't mind you guys doing it as a group, but I'm just not gonna give you the sheet and say, here's all the answers. You guys have to do it. That's part of the learning process. Other things that we look at on here. Fluid. Filtrate. For the first time, we see filtrate in the capsule. Fluid. Plasma, because it is a blood capillary. All right? Everything I'm talking about is on the test. Fluid.
Blood. Oh, it's a tubule.
It's a tubule, so we have filtrate. Filtrate in the tubule. What is unique about this capillary? Came up twice on the exam. Good. Number one, it's got holes in it. They call that fenestrated. Secondly, but you know what else is unique about this capillary bed?
Is the podocytes?
Podocytes, okay, not what I was thinking, but podocytes, surrounded by podocytes, which create filtration slits, good. Third thing, which is a mystery, it is drained by a blank. It's the only capillary bed in the body that is drained by an efferent arteriole. All of those are on the exam. You guys can kind of see how it's put together. The modifications of the glomerular capillary are A, B, C, D. You'll figure out which ones they are, but certainly drained by an efferent arteriole is important. So all of that comes off this slide or is in the multiple choice section of our exam. Number 3, organ. Bladder. Tissue. Mushroom caps that we learn in AP1, that is transitional epithelium, which also is the first question on your exam. The type of epithelial tissue that lines the urinary bladder is blank, and the answer is transitional epithelium. Fluid. Urine. No surprise. Urine. pH. 5 to 8. Specific gravity. There. But you find material in it. What if I told you there was protein in it? Is that normal or abnormal? Abnormal. And its name? Protein or albuminuria. Proteinuria or albuminuria is protein in the urine. It's abnormal. What might cause that?
leaks in the glomerulus?
Leaks in the glomerulus that can be caused by, and I accept multiple answers, infection can damage the glomerular filtration membrane, high blood pressure, which pushes things through the filtration membrane. That's what I mean by give me a clinical term and a diagnosis. I hand grade those, so I'll accept one. Pretty. Just a different picture of what organ? Ureter, yeah. Urine, transitional, smooth muscle, and our good friend, although I don't put this on this exam, you guys remember adipose and the adipocytes from AP1? That's what it looks like. There is a question, not on the slides, there is a question about what is the function of perirenal fat, which is the fat surrounding the kidney,
Anchors lining.
anchoring and structural support. Another picture, organ, kidneys, you guys got all that. The capsular cells, simple squamous, podocytes out here, I won't make you identify it, there's the glomerulus. Another grouping of, you see how nicely organized these cells are? Those are the macula denses. You asked on the other slide about JG cells, and they're gonna be a fine collection of nuclei right next to the macula densa. That's what's DC mean. Distal convoluted tubule. Distal convoluted tubule. Fluid type filtrate, fluid type filtrate, fluid type plasma. That's pretty. Just a lower magnification, a good question, that's the kidney again. Lower magnification, we can see 1, 2, 3, 4, 5, 6, 7, 8 glomerular structures in there. Cortex or medulla? All right, we always find the glomeruli in the cortex. What is one structure of a nephron? Do you know the term nephron? Do you know what nephron means? Nephron is the entire filtering unit, including glomerulus, capsule, and all the tubules. What one structure of the nephron extends deep into the medulla? The loop of Henle. The loop of Henle, so make sure you know that. Those are questions that I just took on the test. What structure of the nephron extends deep into the medulla? And that is the loop of Henle. This is the one you put up. Again, kidney, pretty picture. Those are the macula densa cells there, all right? Tough to see, all right? Looks just like a tubule because of their location. What I said about the JG cells, they're over here. I wouldn't provide that. I wouldn't give you this as a test question, but these are this collection of nuclei over here, the JG cells. So just know if we're next to glomerulus and I'm pointing at a grouping of pretty uniform cells, that's macula densa, and the cluster of nuclei next to those, next to that tubule will be the JG cells. That's pretty. Questions on any of that? Shouldn't be a problem, right? 2 models in back. ID, use your objectives to get at those. All right. I've got my cheat sheet here. We'll just go through the things that you should know. That I can remember off of the exam that I just took. Gland that caps or sits on top of the kidneys is the blank gland. Adrenal gland, all right, easy peasy. 2 kidneys, they sit retroperitoneally In the abdominal cavity, there are 2 ureters. How many entrances, exits does the bladder have? 3. Called a tridome. 2 ureters in, 1 urethra out. I don't remember any question, I guess I'll have to throw it out now that I'm gonna say this, that asked about vascularity, meaning I didn't make you list out the arcuate vein or the interlobar vein or any of the vascularity going into the kidney. You'll need to know renal artery, renal vein, those are the big ones. You'll need to know afferent and efferent arterial as it applies to the kidney. But as far as all of the, this will help you when you're studying this weekend, as far as all of the arteries and veins that are related with circulation of the kidney, they are not on the exam. This is when you guys go, thank you for not doing that. Okay, good. Know the difference between the perirenal fat, the fascia, and the adipose tissue surrounding, that's the perirenal fat surrounding the kidney. Just looking at things up here that I see. This will be where I'll use the model for anatomy. So that what I was just saying about all of these arteries and then all of these veins here are not on the exam. I did put peritubular capillaries and vasorecta. Efferent and afferent are on the exam. You'll need to know the difference between cortex and medulla, no surprise, gross anatomy. Pyramids, columns, certainly anatomically could be identified on the model and back. I also used, there's a section where you have to put things in order of flow, and so if I gave you something like proximal convoluted tubule, papillary duct, distal convoluted tubule, minor calyx, major calyx, renal pelvis, could you put them in order of how urine would be formed and then taken out? So you'll have to go in order on those structures. You can kind of see them up here, but no, the calyces, minor and major, renal pelvis region here, and then all the tubules that we have gone in structural order. PCT, proximal convoluted tubule, has microvilli. Where does most reabsorption occur?
PCT.
PCT, good, because it has enormous amount of microvilli. About how much do we reabsorb in a day? 99% of what? 180 liters is about what we filter. So we usually, I think on this exam, the number I used was we usually create about 1.5 liters of urine a day, depending on intake. If you're sitting there drinking a bunch of water or a bunch of coffee right now, you're probably gonna make more, but about 1.5 liters or 1% of filtrate turns into urine. Know what tubular capillaries are versus what a vasorecta is. They're both capillaries, but the vasorecta will follow that deep loop of Henle into the medulla, and it's for reabsorption. When we get into the microscopy of the kidney, terms that you should be familiar with: renal corpuscle, juxtaglomerular apparatus, nephron. They're all similar, but a little bit different. Of the JGA, juxtaglomerular apparatus, you'll need to know macula densa and JG cells, their function, and what they trigger to release. Important, very, very important. A couple terms just so there won't be surprises for you. I did use the term parietal and visceral. So if you don't remember those from earlier in the year, I want you to look them up, parietal and visceral. Visceral means organ. Parietal's on the outside. Beautiful. That's a scanning electron micrograph. Anybody ever seen one of those before? That's magnified about 10,000 times. You can actually see the pedicels of the photocytes wrapping around the glomerulus. That's an unbelievable picture. All right, then you get to the physio. All of that's anatomy. I mean, just straight up anatomy. I think the anatomy is pretty straightforward. Then you get into the physiology of the kidney system. You can just write physiology of kidneys, what you need to know. There are some basic terms, filtration, reabsorption, and secretion. And you have to be able to tell me where this is happening and what is happening. So when we say filtration, we're moving plasma into the capsule and into the proximal convoluted tubule. You all with me on that? What's reabsorption mean to you? I know we all just said we reabsorb 99%. You got the mantra down, but what's it mean? Where are we moving materials from? All right, that's why we're here. Reabsorption. Lumen of the tubules. Eventually into peritubular capillaries. We're bringing it back into our bloodstream. I said eventually because we actually are just moving it into the extracellular space around the tubules, and then it passively moves into the capillaries. So that's what reabsorption is. So then what's secretion? What is secretion in related to the kidneys? It's from the capillaries into the tubules. And it's a way to get rid of waste material. You guys get that? That's the gist of the exam on physiology. You have to understand these terms and where things are moving from and to. Factors that regulate the first one, which is glomerular filtration. These are the three. There's a couple of questions on the exam where I changed them. For example, what if I told you the capsular hydrostatic pressure was doubled? What would that do to filtration? Capsular hydrostatic pressure is doubled, so make that 30. It's a negative pressure. That's a negative pressure. That's a positive pressure. Hydrostatic pressure is doubled. We now have negative pressures that total 60 versus blood pressure, which is 55. Filtration stops. You guys get that? It's a calculation. This is a positive. These two are opposing forces. We always want to have a positive. Blood pressure needs to be high enough to move the filtration process forward. If I told you that capsular hydrostatic double, filtration would stop. Is that a good thing? No. So make sure you know these 3 factors of glomerular filtration. What's the biggest one? And the one under our control? Blood pressure. And that's why the kidneys are so important in monitoring and adjusting our blood pressure to make sure we have enough pressure that we can filter the blood. And blood pressure is the big thing on this exam, so make sure you know glomerular or blood pressure are the factors that the kidneys have to regulate that. And here they all are. This gets back to our worksheet a little bit. These are all in your notes. This is the big bad boy on this exam. You can kind of figure it out, but we have low blood pressure. When you have low blood pressure, what's that gonna do to filtration? Slow it down, we're not gonna have any, all right? That's not good for our body. So all of these factors in this chart will increase GFR, glomerular filtration rate, and I gave you several mechanisms of where these can actually happen. So we've got these over here, which are intrinsic, and then we've got the extrinsic, and a lot of in the green here, you'll need to know for sure, because they all raise blood pressure. Make sure you know what angiotensin II does. Make sure you know what aldosterone does, all right? Make sure you know what ADH does. Those are 3 important chemicals on this sheet that all help us to increase the filtration. Sympathetic nervous system can do the same thing. Remember I told you when we started this discussion, there's glomerular filtration, there's reabsorption, and there's secretion. And I told you reabsorption takes place here from the tubules, eventually back into the capillary. That's what reabsorption is, all right? 2 things. I didn't actually ask you anything about what moves passive or actively in our system, but what's the difference? I did ask you a question on what's the difference between passive and active transport. Anybody remember? Passive is high to low, does not require energy. That's what you need to know. High to low does not require energy. Active transport requires energy and usually goes against the concentration or electrical gradient. Are we good on that? But I didn't ask you, does glucose move passively or actively? So you don't have to go back and memorize each individual solute as to how it moves. Except for water, and I heard it, Eli. How does water move? It moves by osmosis is the process of water movement, and that one did make the test. That was the only one. While we're on that topic, 2 topics under movement of water that you should look up and be familiar with. 1, obligatory water reabsorption. It's in your notes. Obligatory water reabsorption. And 2, solvent drag. Those are both in your notes and in your textbook. I would look them up. That makes that question pretty easy.
Solvent drag?
Solvent drag. Water follows what? Sodium. Sodium's under the control of what hormone? Aldosterone. You have to get those down. That's when I take the test. I look at that, I go, well, that's easy. Well, you guys have never had aldosterone before. It's easy for me because I've had it for many, many, many decades. But aldosterone causes reabsorption of sodium, water follows. Kind of covering fluid and electrolyte at the same time with the kidneys, because I'm looking up here, I'm going, oh yeah, I did ask that too. If sodium is high extracellularly, what is high intracellularly? Potassium. We learned that in AP1. It's a review, but it's on the exam. All right, that has to do with fluid and electrolyte. High sodium outside the cell, high potassium inside the cell. look familiar to you? This is the countercurrent mechanism. I told you that you don't have to know it because all these milliosmolarities of the filtrated is moving through are pretty irrelevant. You'd memorize it and forget it, and you should. The only thing I did ask about is do you guys know where we're at? Here's the capsule, TCT, loop of Henle, this is descending, this is ascending, distal convoluted tubule, collecting duct, papillary duct down here. Fluid inside, filtrate, fluid down here, urine. First drop off of the papillary duct is actually urine. The only thing I did want you to know here is that most reabsorption occurs in the PCT. Water is reabsorbed in the descending loop. Sodium and chloride are reabsorbed in the ascending loop. That'll help you with a few questions.
Yeah, can you say that again?
Oh, I'm just making stuff up. Of this nephron, this is nephron. PCT is where most solutes are reabsorbed. Descending loop is for reabsorption of water. Ascending loop is for reabsorption of sodium and chloride. Don't have to know the numbers. You'll thank me later. I showed you this picture, and you guys would be studying and probably go, wait a minute. He said that most stuff gets reabsorbed in the proximal convoluted tubule, which is here, but look over here, E, collecting duct. Does it look like a lot gets reabsorbed in the collecting duct? It appears like it, but in concentration in numbers, a lot more here. That's possible to do these here, but that's only like the large intestine for homeostasis. Only a little bit actually moves. This is ammonium. Came up on the test a couple of times. You ought to be familiar with it. It looks like it's being secreted or reabsorbed. It's being secreted, which means we're putting it from the extracellular fluid back into the tubule because it's about to form urine. And it's a way that we can get ammonium ion out of our body. Do we want urea back? We don't. It's a waste product. You should probably know your waste products that we try to get rid of, like urea, uric acid, keto acids, creatinine are all waste products we try to get rid of. This will help you with a question. Waste products. Nothing on urination, micturition, no sphincter. Fluid electrolyte. The basic things I touched on this exam, like the difference between intracellular fluid and extracellular fluid. Can you tell the difference? All right. Where is most of the water in our body found? Intracellular or extracellular? Inside the cell. Inside the cell. Most of our water is inside the cell. If it's not inside of the cell, it's outside of the cell. They call that extracellular fluid. And we talk about several types which are on the exam. Things that are extracellular fluids that we've studied this year include CSF, lymph, plasma, bile, pancreatic juice. Know your compartments, know what's in them. This is important. When we get to system, fluid and electrolytes, all about basically maintaining pH. What two major organ systems we've covered this year are our biggest regulators for body pH? Kidney, number one. Respiratory, number two. Those two. Those are our biggest modulators of pH in our body. It's a test question, now you have it. Piece of cake. Some people tend to like to put digestive down because we talked about hydrogen ions a lot in digestive, but it's not a way to get it regulated in our body. So it's respiratory and renal. Two major hormones. One, ADH. Antidiuretic hormone. Couple questions on ADH. First, what inhibits ADH? What inhibits it? Had a baseball meeting again last night at office. Had a beer,
Oh.
coffee. Those are inhibitors of ADH. That causes you to diurese. Know where ADH comes from? Oh, it's target tissue, Shailene. This is Shailene's form that she put up on Moodle. There's the target tissue collecting ducts, and there's the effect on the target tissue, which is increased water reabsorption in our system. You should know osmolality or osmolarity, same thing for our exam, and what that means. The second major hormone, you really have to know these two hormones well. These are the big ones. Angiotensin 2 is important, it's on Shailene's as well. Renin is important. Aldosterone and ADH are the two big ones. They come up 2 or 3 times. There's aldosterone. Aldosterone. Released from adrenal cortex, targets the kidney tubules. Anybody know specifically where in the kidney tubules? I'd like you to know a little bit more about, just not kidney tubules, but they are specific. Descending loop of Henle and the DCT, target tissue for aldosterone. Increases sodium reabsorption. When we bring in sodium, we kick out potassium. What follows sodium? Water. And anything in water follows. This chart comes from your textbook. You might go, oh, do we need to? Yeah. This is the whole big picture of the physiology of the kidneys. It's actually out of the fluid and electrolyte chapter in your textbook. I told you not to look at it, but you might need to actually find some things out, because this one has renin, angiotensinogen from the liver. Have you guys heard of that? It's on the test. Angiotensinogen. It's converted by renin into AG1 into AG2. And AG2 effects on peripheral resistance, increases blood pressure, affects aldosterone. All of this really is a nice chart. There's ADH, Everything on there is relevant for our exam. Shailene, this is where a lot of your information can come from, or should have come from, because this chart out of fluid and electrolyte has 5 major chemicals on them that are listed on Moodle as things to know. Are we good on that? All right. Do these things up here all increase blood pressure or decrease? They all increase, because that's what we're trying to do. If we don't have blood pressure, we don't filter, period. So everything in our body, except for one One little thing that comes from the heart is a decreaser of blood pressure. Everything else in our body increases blood pressure. That's an important little chart to me. Yay. Nothing. See all this stuff at the bottom? All of this secondary, simple to view, how things move, and I told you, do not memorize that, all right? Know what passive and active is, but I'm not gonna ask you specifically, how does phosphoric acid move, by primary, secondary, etc. The only two things we might put down, let's put them down, not might put down, you better put them down. You'll need to understand a couple of terms that I don't think I covered in the notes, and I put them on the test, and I thought I better bring them up. One is transport maximum. Transport maximum. So transport maximum is a condition where you have these carriers here. And if you have an enormously high amount of a substance or solute in the filtrate of a tubule, can you get them all on the carrier? No. So if you have a very, very high concentration of something, a poison or a chemical or even glucose, and the glucose is really, really high, would you expect to see glucose in the filtrate? Is it small enough?
Yeah.
Yeah, glucose is in the filtrate, it's small enough. But don't we want to reabsorb all of our glucose? Glucose is not normal in the urine, yet it comes out in the filtrate. But there are times when our transporters are full. That's called transport maximum. People with diabetes, for instance, often reach transport maximum because their glucose is so high that they can't transport it all. They've reached transport maximum, and it is then found in the urine. When you test urine and you see glucose in the urine, do you think diabetes?
Yeah.
Yeah, that's usually the first thought. That's because they've reached transport maximum. It's called TM, transport maximum. It's on the exam. And therefore, you'll find glucose in the urine. That's called renal clearance. That we're actually clearing stuff out of our body. But that is not something we want to clear. There's a drug, well, like, renal clearance is on the test. This drug is not, so don't worry about it. But I'm just telling you that sometimes clinically they want to test to see if your kidneys are working. So you ingest this drink that's called inulin. Looks like insulin, but it's inulin, and you drink it. And it goes through your body, goes through, and they will start capturing your urine. And they want to see that this inulin goes through as filtrate and comes right out in the urine. There's no transporters for inulin. So you drink 100 milliliters of inulin, and 100 milliliters of inulin come out. Does that mean your kidneys are clearing things? Yes, it does. And so they test for that. If some of the inulin remains in your body after an hour, then you know that the kidneys are not cleaning your blood properly. So it's a test for good renal clearance, which is the term on the exam. Renal clearance, just moving things out of your body to the kidney. Two questions on these. Make sure you can compare and contrast alkalosis and acidosis. And compare and contrast the difference between respiratory and alkaline. Metabolic, respiratory and metabolic. Acidosis and alkalosis. Hey, if it has CO2 written next to it, CO2 levels, you know that you're right here in respiratory, all right? Because metabolic has nothing to do with CO2 levels. So if you see CO2 levels on the test, you immediately can go respiratory. Then you have to figure out whether it's acidotic or alkaline. Another term, when we were talking about osmosis, just make sure you're comfortable with these 5 terms. Number 1, permeability. These will just make questions easier. I don't ask you, permeability means, but I use permeability in a possible answer. So permeability, osmosis, osmolarity, solutes, and solvent. Covered it all. Any questions? About 58 questions-ish, and then practical. Take you about an hour or less. All right. Monday, fill out that chemical sheet. That'll be important. Renin, aldosterone, angiotensin, ADH. Let's get that done. I'll check in on it on Sunday, make sure it's done complete and correct. See you at 8 Monday. Got nothing else. I can't believe next week from today, we're starting the final exam in this class. And it's fun. Reproduction